start. So first of all, welcome to everyone and thanks so much for being here in our research seminar. I'm very, very happy and thrilled today to have Dr. Eddie Watkins with us, giving a talk and also visiting. We just went the last 3 days with our other collaborators and research associate here at Lisa George. We went to Ink Lake, State Park, Colorado Bend, and Longhorn Tavern collecting ferns. And Eddie, you might just know what he's doing here after the talk, and he's also interested in doing adaptive ferns now. So it was great. And so just a little bit about Eddie. He is a professor at Colgate University in New York. He's the director also for the Center for Learning, Teaching, and Research. He's a Southern Botanist. He has a bachelor from
Auburn University,
where he worked with John Freeman. Maybe some of you are familiar with him. Then Eddie moved to do his master's at Iowa State University with Don Farrar, working with the lymph nodes with the shield burn systematic. And then he went to Florida, University of Florida, to do his PhD in fern gametophyte ecology. And finally, he got postdoc at the Arnold Arboretum in Harvard University. So I have met Eddie long time ago. I was a student, I was a baby, I was already a friend. Had a course in Costa Rica that I took as a student in 2008, and he was teaching it with Roy Moran, who was at that point my PhD advisor. And now we teach the course together in Costa Rica every 2 or 3 years. And so I'm now a teacher. We are really good friends and colleagues. I'm just very excited to have Eddie here. I'm not going to take more time so you can share your experience with you. But thank you so much, Eddie, for coming.
Thank you.
Thanks.
Thank you. I'll give you a hug. Thank you. Okay, thank you, Alejandra. You know, I had this talk put together before, and I showed it to Aleja last night when we were on the way back, and she said, "No one wants to hear about boring physiology, Eddie. They talk about your new stuff." And so this talk, literally, I just dumped the whole thing and started rebuilding it last night. So I'm not going to talk about the cost of doing business in ferns, although if I were to do that, there were 106 slides in here. I deleted like 80 of them so I could give this talk. So, so the talk has changed a little bit, but we'll have fun nonetheless. It's really happy to be here. I've known about Britt for a long time. When I was an undergrad at Auburn, working with John Freeman, he was a trillium expert. I don't know if you know who John was, but you know, it was a time when I was learning about herbaria and collections-based research, and I worked on Botrychium lunarioides back then. We finally published. I started this work actually in high school. I was a real nerd in high school and started looking at lunarioides in high school, and worked with John on a publication we eventually published on that. Just the classic old new distribution and new county records of lunarioides in Alabama. So I was just visiting some of those old friends in the herbarium. So really happy after all these years. The first time I heard about Britt in '98. I don't know when Britt started, but I heard about it back then, and it's the first time I've been here. So I'm a little embarrassed to say that, but I'm very happy to be here and happy to to see a little bit of Alejandra's life. We've been friends for a long time, so it's nice. And you're all very lucky to have her, let me tell you. So congratulations to you all. We'll see where this talk goes. This is a brand new talk for me. I have never really talked about this stuff, so we'll see where we're going to go. But I want to first start out by thinking about ferns in a way you probably haven't thought much about. And I really study reproductive ecology of ferns. But when I say that, I study the sexual phase, the gametophyte generation. That's primarily what my research has been on. Not on this boring, stage, the diploid stage, but most of my career has been spent on a lot of things. But when I think about ferns, I think about the independent gametophyte generation. And I'm going to share some of that work today, specifically on sort of the physiology of fern sperm. Okay. And so this is what your typical cordiform fern gametophyte looks like. I always tell my students, this is a sexual phase because it's shaped like a heart. So there's all love. And so But it turns out fern gametophytes don't all look like that. As a matter of fact, most of the gametophytes don't look like that. There's tremendous diversity in form, from these sort of thallus type flattened gametophytes to the classic cordiform shape, strap shape. They're really variable. And so a lot of my early work was trying to understand sort of form and function relationships. Why do fern gametophytes have such modified, crazy morphologies? And is there a function to that morphology? And we've done a lot of work on water stress tolerance and nutrient relations and light stress. And there are some reasons why, but I'm not going to talk about that work today. I'm going to talk about some other stuff, really focusing on the biology and ecology of sperm physiology in the beginning of the talk. And so at least you think that, fern gametophytes are this big mess and you can't identify them. Let me tell you, in Texas, they're hard to find. We found one little tiny thing yesterday or day before, whenever we were up at Inks Lake. They do have variation in form and they do have some characters. So here's a good example of Cyathea multiflora. This is a cordiform gametophyte here, but see these multiseriate hairs right here? Only Cyatheas do that. And so there are not a lot of characters in gametophytes, but they do have some. So it is possible to identify them. But the work I'm going to talk about today is really going to be on, stuff that we've grown and reared in the lab, okay? And so I'm going to talk about fern sperm physiology, really focusing on longevity, competition, and velocity. So ferns, as you know, sort of plants, green plants in general, share a common ancestor with green algae, maybe the Zygnophytaceae now, or whatever group it is. But they originally share some common ancestry with their aquatic environments. And they still share some of these characteristics with the fact that they still maintain this motile aquatic swimming sperm. And so these are, I just showed that video again, just for fun, or for fern. There it is. So these are all the spermatozoa that have been released. These are a bunch of antheridia down here, the male sex organs releasing. There's one, their sperm are being released in these little mucilaginous capsules, and then they dissolve and they just will go off and swim around. And so not many people have actually attempted to study this because it's actually quite difficult to do, a lot of labor. But so I want to talk about some of the work, and this work is very preliminary. And then I'll talk about some of the work we've actually published on this and hopefully tie those two stories together. I hope I can do that. So what are fern sperm? What do they look like? What are they made of? How do you begin to think about them? This is some incredibly important work that's done by Karen Renzaglia and Renee Lopez Smith at Southern Illinois. She did a lot of work with fern sperm, spermatozoa, plant sperm in general, looking at their sort of morphology and ecology. There's not much to them. These are antheridia here. You can see the cap of the antheridium, and inside of these antheridia are the individual spermatozoa in there, and they have been released either in these compact sort of spiral forms, or they can become elongate after they get kind of tired. We'll talk about that in a minute. And here's what they are. They have some flagella, and they move around and spin around with this flagellar structure, and have a little ball of cytoplasm here and a one single mitochondrium. So they swim around, and as they get close to fertilization, they actually drop off this cytoplasm for some reason, and then they have a little bit of like a half of mitochondria left. There's not much to them. They're very small, and we don't know much about. In fact, before we did this work, we knew nothing about how long they live, how fast they swim, nothing like that. But it becomes really important to understand the physiology of the sperm because it controls so much about the reproductive success of these plants, because they are motile swimming. And it's not just simply, if you have a question like how fast do they swim, how long do they live, et cetera, there's real implications for that kind of knowledge, because it's not like the sperm swim from one gametophyte to the other in this straight line. These are very small parts of the life cycle, and they actually swim these very complicated matrices. And so this is, for example, a cartoon of a moss mat and a fern sperm trying to find its mate. And so they have to swim in these very complicated patterns through soil, around soil. So you can imagine that it's not an easy life that these things have. And so understanding how long they live and things can be very important. So the questions I'm going to try to address today is, how long do fern sperm swim? How fast do they swim? And is there any evidence of competition or intraspecific interactions with fern sperm? Okay, so let me just ask you, given what I've told you, this is actually equisetum sperm, but how long do you think fern sperm swim? What would you say? How long do they swim? 24-hour a day? Yeah, a week? That's very ambitious. But before we did this work, most fern biologists and most folks, they just a couple minutes. That's all they do, because they have not much to them. And so we went into this work trying to understand that with the idea that these things don't. I didn't believe that necessarily, but that's what the common conventional wisdom was. And nothing was published on this at all at the time. So we did a lot of different things. I'm going to share some of the work on some of the tropical ferns. Went down to Costa Rica, collected lots of spores, brought all these things back to the lab, grow them in an agarose medium, which allows you to grow these things very quickly, and you can see them very easily. And we grew these things up in these biochambers in these controlled environments. And the challenge with ferns is, contrary to that life cycle that I showed you of a single spore giving rise to a bisexual gametophyte that have both male, make egg and sperm, not many ferns actually do that, okay? And so that's total error in the textbook. So we've kind of corrected it in some books. But when you sow spores in what we call multi-spore culture, this is an agarose plate, and you sow a bunch of spores in there, they almost always become female. And so it's a bit of a challenge. So when you do that, they become archegoniate and produce all these archegonia, and they're not really bisexual. They start out mostly as female. They may eventually become male, or something may happen to them, but they almost always become female. So then you're challenged, if you want to study anything about the spermatozoa, you have to create males. And we're fortunate, at least in some cases. Am I going too fast, Alejandra? I do tend to talk fast. She's always correcting, Eddie, slow down. I get too excited. So you can make males in some species. So ferns have this really interesting sex determining system. So in many species, the spores land, they germinate, they become female, or before they become female, they begin to produce this chemical, excrete this chemical from their meristematic region called an antheridiagen. It's a gibberellic acid type derivative, if you know about plant hormones. And what this does is this hormone will ooze out of these gametophytes, pre-female or female gametophytes, and they chemically induce everything around that female to become male. And so she, in a sense, can surround herself with male gametophytes. And that's what's indeed happening here. So we utilize that system to make males. So not all species have this antheridigen system, but many do. And so what we do is we grow up gametophytes, and when they get to the certain point, when they start making archegonia, we put them in new plates, and we sow spores around them. And these things will become what we call precocious males, just covered with antheridia, and that's all they do. So they become males. And now we have, what we need to do the experiments with sperm. And so there are a couple of different ways to do this. This is an example of, for example, here's a cartoon of a gametophyte surrounded by males. If the species has a system, it can respond to its own system. So you have a gametophyte of itself, and it turns its own gametophytes to males. Or there is some exogenous production where you can, for example, Pteridium has a very strong system, and it induces a lot of other species to become male. So if a female Pteridium gametophyte will begin to grow, and it has a very general type antheridigen, and most things around it will either be stunted or it will turn them into males, even if they're not related to Pteridium. And so you can harness the power of Pteridium to create males, okay? Because they're not so easy to find. So that's how we sort of designed this in order to get enough sperm to do this. And so we have about 30 some odd species in this data set now, but the ones that really worked for the full data set this is brand new stuff still coming out now, but these are all taxa from Costa Rica. Pteridium aquilinum, that came from Arizona, I guess, but the bracken fern, Pteris propinqual, which is this thing right here, a big pteris. You might be familiar. Pteris vittata must be here in Texas, I guess. What is the common name of pteris? Brake fern or something like that. Salpiclina volubilis, which is in the genus, the family Blecnaceae. It's a blechnum. It's a big, beautiful climbing, vining ferns. These things can become 30 or 40 feet long, and they climb up into the tops of these rainforest trees. Hypolepis repens, which is this thing, Dinstadiaceae. Is that right? And so I don't know what you have here. Dinstadia, hay-scented fern up in New York. We have that. Serpicolon tricereale, the Polypodiaceae. There's no common name for that I know of, but it's an epiphyte. And then Oleandra articulata, which is this simple leaf thing, grows way up in the top of the canopy in these trees in Costa Rica. I think it's Dropteridaceae. Is that the right family? Oleandra? Oleandraceae. See, she's the taxonomist. I'm the ecologist. And so we have a diversity. We intentionally wanted to sample a broad phylogenetic sampling to do some cool phylogenetic analyses, but we're still working on that. Well, I won't be able to show you any of that. I'll keep it fairly straightforward. So those are the species we worked with. And we did a lot of different things I talked about how we grew them before, but we took the mature gametophytes that had antheridia on them, put them in a drop of liquid medium, the same medium that we grew them on, but without the agarose. And we put them on microscope slides, depression slides, and we observed them. The question we had is how long they live, and how fast they swim and all those questions. So we had to set it up. And so we had all these microscopes set out with all these depression slides on it. And we would turn on the light, do some measurements, turn off the light, put it back in the bag, and try to figure out a way that we could see how long they were swimming and for how fast they were swimming. And then we used this software to analyze this, do some video analysis to look at how fast they were swimming. So here they are. Here's an example of Salpiclina volubilis, one that really participated very well in this experiment. So this is what a lot of students spend many hundreds of hours analyzing these videos. And what we did, we set up the microscope camera, and we had this Logger Pro Vernier software running in the background. We tracked each sperm for about 20 frames at 6 frames per second. And we only track them when they're in focus because they do swim up and down, and we want to know how fast they're swimming. If they're going down, it looks like they're swimming slower than they are. So we tried to be as careful as possible. Not the greatest technique, but it worked pretty well. And then we calculated the velocity between each frame and then averaged over that entire 20 frame interval, if we could get 20. And you'll see this is what it looks like. Here's the sperm here, the student tracking it frame by frame. There's another one there. So she would make these videos, and then we would sum up. We would take the mean of this entire track here of this sperm. And so it took a lot of time to go this fast now, but she would go click, 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 click for hundreds of these videos there's a little sperm coming in there. There's another coming through. So yeah, it's kind of fun. So those are the spermatozoa and all over the place. Okay. And then, so we did that. And then we also wanted to understand, it was not only take those data and do some velocity calculations, but look at, how long they lived. And so this is kind of complicated because it's a fairly artificial environment, but we wanted to understand, how do you begin to look at, you can't find the same sperm over and over again. They're so small. So how do you do this? And so what we would do is over a time period, we would, I can't remember how many we did with this species, but every minute or 2 minutes or whatever, we put the slide on there and we counted how many were still alive, up to 10 total. And you see, so they do, their mortality goes pretty quickly, but you see they actually, here's when they first are released. Here's that, I keep pushing the wrong button. This is Salpiclina at time T0, and this is after an hour. So they're slowing down, and they're not nearly as many sperm swimming around. So we tried to figure out a way to be able to look at speed, and also speed in the beginning, at the end, and how many were still alive. And so in some of these data for the absolute longevity, we're looking at maybe just a handful, 2 or 3 spermatozoa that are still alive. So it's the maximum possible longevity for this study. So how long did fern sperm swim? Here we go. So this is longevity in minutes. And these are the species across the x-axis here. Gray and white, that's for another thing I was going to talk about, epiphyte versus terrestrial. Don't worry about the colors. So what you see first is a couple things. These are with the self-endogenous experiments. We had the same species gametophyte female with the same species spore sewn around it. So no interactions outside of that species. And there's variation across the different species here, right? So Oleandra articulata had the slowest swimming sperm. Hypolepis repens had the fastest swimming, the oldest sperm. Sorry, this is longevity. So they lived about, what is that, about an hour or so. And these lived almost, a little bit more than 160 or some odd minutes. So this was it seems pretty simple analysis, right? But this was actually pretty big, sort of a big discovery in the sense that most folks thought these things could survive for a few minutes. But they can at least survive for hours, which was a really big deal. So that's a first huge discovery. There's more to it than that, obviously, but they could swim for hours. And that, we had a lot of interesting discussions about sort of these data. And some of the work we're doing now is looking at, okay, well, you've been swimming for an hour, but can you still fertilize an egg? And that's been really, really hard to do because trying to pipette the sperm off and put them on the females, and mostly it's failed. But so I don't know if you swim, maybe how you can swim for an hour, but if you're worn out, or if you lose your capacity to fertilize, then what's the point? It's not, so we're trying to work on that question right now, which has been really challenging to do. We only have one species that we have any understanding of, and I don't believe the data right now, so we're still working on that. But they swim for hours. So now you can take that home and talk about your next cocktail party. So how fast do they swim? You saw them there, right? And some of those videos were slightly slowed down. And so that becomes an important thing, but you're going to see a really interesting connection in a few minutes when I get to that slide. Swim for hours, how fast do they swim? And it matters because there's a relationship between survival and swimming speed in just a minute. But it is interesting to understand, is there really what we're trying to get at is there variation across different species? Does that ultimately relate to their ecology, or their physiology, or their reproductive success? So there's bigger questions behind this. But here we have velocity here, right? So this is species across the bottom, as you'd had before, and this is in millimeters per second. So Oleandra had the slowest swimming sperm. Hypolepis repens had the fastest swimming sperm. You might start seeing some connections already here, which is very interesting. So you're, okay, I don't know, Hypolepis, the winner, this is the Mario Andretti of fern sperm in our study, about 0.25 millimeters per second. What does that mean? So little data out there. There's a tremendous amount of data on human sperm and animal sperm and all, you know, I actually reached out to our local hospital and how do I begin? Because they have all these complicated lasers to measure human sperm fertility. And I said, can I, you know, come and do this work on plants? And they were like, what are you talking about? And I even had one of our librarians, we were working this out. I wrote her an email, saying I have the student wants to study the sperms project, and she wrote back saying, I am not studying student sperm. I'm like, no, it's plants. People have no idea they do this. And the hospital even didn't want, so they wouldn't let us in for obvious reasons, but private fertility clinics. But they do have different velocities. So what does that mean? Well, here's a good example of moss. The fastest, the swimming moss that I could find is there are only 2 papers I could find with anything about sperm longevity, is about 1.3 millimeters per second. I'm not quite really willing yet to say ferns are slower than mosses, but there's just not a lot of data out there, right, to show this. So what does this mean? I did some back of the envelope calculations for this. I got to get to the airport today. So this is Brit to DFW. It's about 27.9 miles according to Google. And so that's about 44,000 or so millimeters. So if I were going to take, I'm going to take Hypolepis repens to the airport, it's going to take me about 49,000 hours to swim that far. So they don't, you know, they don't swim very fast, right? But what we don't yet know what the sort of ecological consequences. What does that mean? Is it probably fast enough? Because they're, you know, ferns are highly outbred, just like any other group of plants, and so they're obviously reproducing sexually on the landscape. So I don't know, not so fast. We don't know what to compare that to yet. So hours, it's kind of slow swimming things for hours. Okay. Any questions or comments about that? You can interrupt me at any point. Yes.
What was the average sample size for tackle?
So most of these were, so we had 10 individuals and we had 10 spermatozoa per individual. So about 100 sperm, you know, all total. And, you know, there was some very limited variation, surprisingly, across the sample size. It's about 100, I guess, sperm. Yeah. Yep. No. Yep, and yep.
Is there any data like on why that is, or
you know, I think some ferns become bisexual right away, but most of them a lot of them will become female first. I mean, so yes, it has something to do with the fact, this gets pretty technical, that ferns are homosperous, meaning they don't have sex chromosomes like some mosses. They don't have male and female spores, except for the aquatic ones, like many lycophytes. So a single spore can reproduce to the sexual phase, which is the gametophyte. And that gametophyte produces sperm and egg. And the sperm and egg are the products of mitosis. In our bodies, meiosis gives rise to gametes. But in plants, that doesn't happen. Mitosis in all plants gives rise to the gametes. So if you have a sperm and egg on the same structure that are produced through the product of mitosis, mitosis makes identical cells. Then if that thing selfs, then you have a sporophyte that is completely homozygous at every locus, right? So it's technical because it gets at what the definition of a plant. But because plants have mitotically derived gametes, there's some dangers not dangers, but if you create a homozygous sporophyte, if you have a single recessive lethal, it knocks it out. So I think in a sense to avoid that, ferns, part of the superiority of ferns is that they have this life cycle. They can land, become female, hang out. If they're not for life, they may become bisexual. And if they can self, hey, at least you did something. You may have no genetic diversity, but if you're there for the long term for 10,000 years, then you maybe rebuild that. That's a technical, yeah. But that really gets when I ask my students, what is a plant? It's not about boring cell walls or chlorophyll. is really about the fact that how they reproduce and the fact that mitosis gives rise to gametes, not meiosis. And that makes it, you got to dig back to like high school biology to remember that stuff. But okay. So one of the things we kind of picked up when we were doing this study is that there seemed to have been some interactions between, interspecific interactions with sperm. So what we did see, we did learn that velocity and longevity varied significantly across some taxa in these multi-spore cultures. But when we started looking at the endogenous, the self, when you're looking at species one on species one, but when we had pteridium in there, we noticed that in some cases the sperm were not behaving like they did when they grew by themselves. So the question is, this is a potentially competitive matrix. What are the gametophytes doing to their potential competitors? We know that they can transform them to males, and that can cut them out because you have to have the egg. But if I'm the early growing female gametophyte, everything around me becomes male. Even if they're not my same species, I prevented them from reproducing. But what happens to the sperm? Is there any sort of are there interactions with the sperm? It turns out we found this crazy result, which we were totally not expecting. And it kind of sunk a lot of the work we were doing because we were using pteridium to make all these males. And what we found, and here we have just velocity, for example, and here we have the species. Black is when they were growing with pteridium. So this would be pteridium, and this would be salpiclina, for example. This would be salpiclina with salpiclina. When we grew them with pteridium, in some cases, here's Oleandra reticulata. With pteridium, the velocity dropped by more than a half. So just by growing here like this, when these sperm were released, they actually swam about half as fast as they did when they grew by themselves. And that was the case with Oleandra, salpiclina, and Serpicolon, these three species here. But in some species, it didn't seem to matter. Maybe a little bit faster, but not significantly so. So there's real potential for some sort of interactions at the level of the sperm. So that growing with this presence of this pteridium is somehow negatively impacting the velocity of my sperm and longevity, which I'm not going to show you. So this stuff is so, I love this kind of work because, we get so caught up in this crazy molecular biology and using pipettes and there's so much fundamentally important biology we can learn with just a piece of string and a ruler still. And this is just looking at sperm swimming around. It's so basic, but it's so fundamental in changing the way we think about the way these plants operate. And so, gosh, I don't have a lot to say about that other than the fact that, it does have an impact. And not only with the velocity, I guess I will show you longevity. Here's Salpiclina volubilis growing by itself, right? You see velocity here. The sperm slow down over time. Here we have time on this axis. Look at when you grow it with pteridium. It just, not only they swim slower, it basically is killing them. But look at Hypolepis. I mean, there's some differences here in some places, but not that different. There's so much stuff we still don't know about the way these data are coming out, but there's a lot of interactions happening at that level that have a fundamental, can really change the way we think about these plants. We do have evidence of competition, but there's much more to come with that. I had a couple of slides I put together, but I don't want to get myself in trouble because there are a lot of half-baked ideas. And so I didn't want to share that, but there's a lot more stuff coming for this part of the study we're doing. Okay. Now this is something that was still trying to puzzle with, in that we did find a very interesting relationship between longevity and velocity. These are the species here. I didn't put the species name on it, but you could probably guess who that is. Right? Guess who that is? That's Hypolepis, and this is our Oleandra. There's a fascinating relationship between longevity and velocity. So what I mean by that is that if you're short-lived, you swim pretty slowly. But if you're long-lived, you swim faster, right? Or vice versa. I don't know what's causing what, but you live fast, die old. The ones that swim fast live the longest. I don't know if they have a different type of mitochondria. We don't know anything about what's going on. But when you do this kind of research, you get like an R-square of 0.97. It's like something's going on here. Maybe we messed up our data. My first with the students, I went in and said, "You've messed something up. I don't believe this." I had them do it again and reanalyze the videos. But there's something going on here, and I don't know what it is. I've talked to some of my biochemistry and physiology friends. It's not entirely clear yet. So we're working on expanding this, and many of the other species we have fall along this line, although not quite as nice as these two. So there's something also fundamentally going on related to velocity and longevity in fern spermatozoa, which is, remains to be seen. Okay. Now let me see how I'm doing on time. So, okay, good. We got through that in half an hour. Good. So now how can we use these data? This is awfully esoteric. So I wanted to try to talk to you a little bit about how we have actually, why these data might be more interesting than just the curiosity at a cocktail party that we talked about earlier. So I want to talk about some work we've done now with Gyropters. This is a paper, none of the stuff we showed before was published. These data are all published now. If you know the wood ferns, if you know anything about ferns in general, you know they're very promiscuous. And this is a good example. The Gyropters is the most promiscuous of anything. I mean, to give you an example, so these are all sort of the different hybrids and interactions that Gyropters, the wood ferns have. Here's the marginal wood fern Gyropters marginalis. It forms 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different hybrids with different species. It's nuts, right? So they're very promiscuous. Ferns have a lot of capacity to make hybrids and to form all these really interesting interactions. And there's some that are well known and others that are less well known. And so what we do know about hybridization in the wood ferns and ferns in general is that it can be asymmetrical. And what I mean by that, I pulled out our southern friends here. Gyropters ludoviciana, which is in the eastern part of Texas, down in Alabama. It's the southeastern thing. Gyropters celsa, which I think there's a single population of in Texas, on the way out eastern Texas. And then Goldiana, which is a northeastern thing. These are two diploid parents, and they form this tetraploid Gyropters celsa, okay? So you get the complete genome LL from ludoviciana and the complete genome from Goldiana. So it's a big fertile tetraploid hybrid. And in almost all cases, what I mean by asymmetrical, when they form this, almost always Goldiana is the sperm donor. Not always, but it's almost always, and ludoviciana is always the egg donor. There's asymmetry in a lot of these hybrids, the way they form, which is curious. We've known about this for some time, but we didn't really know why, okay? And so we set up this study to examine two sort of triads of hybrids. And we worked on Gyropters intermedia, a very common intermediate wood fern up in New York, probably the most common fern in the Northeast, wood fern for sure. Gyropters carthusiana. This is a diploid. This is a tetraploid. They form this triploid hybrid called Gyropters triploidea, which is convenient. And then intermedia also crosses with cristata to produce this triploid called bootii, cross bootii. Why do I say these? In many cases, these hybrids are more common than their parents in some of these habitats. It's really interesting. They're very abundant. And so here they are. This is what they look like in the parenchyma. Here's carthusiana. Here is intermedia, the tetraploid and the diploid. They form this triploid sterile hybrid triploidea. Here is bootii, cristata, a swamp thing, intermedia upland forest, and bootii. They're pretty easy to diagnose these things in the field. Actually, it's not, when you see bootii, you know it. Like this is a weird bootii. And it's very, very common. So we didn't know about these. We assumed there's some asymmetry here. It hadn't been studied before. So the first question is hybridization asymmetrical in these taxa? And if it is, why? Okay. And so we sampled plants across Vermont, New York. My former student and colleague, Wes Testo, did some work on sequence the chloroplast gene. We sowed the spores of the diploids, obviously, the diploids and the tetraploids. We measured ferns. We're going to try to link this back to fern sperm. That's the connection here, sperm physiology. And then we measured some archegonial characters. Okay, so we're trying to understand why. Maybe there's something going on with the sperm or the archegonia or something, so the gametophyte generation. This is what we found. So if you know anything about plants, you know that the chloroplast is maternally inherited. So if you sequence the chloroplast genome of ferns and you have a hybrid in there, it's going to be paired with its mother. So you can tell, if you know a hybrid, you can run these chloroplast phylogenies and you know who the mother is. Okay. So in this case, this is triploidea. This is bootii, the sterile triploids. The green is carthusiana. The orange is intermedia. So what this means is that 86% of the individuals we sampled of triploidea at these different populations here, 86 at Basic Creek, the mother was carthusiana. What that means is 86% of the time, the father was intermedia. So overwhelmingly, intermedia is always the sperm donor in this triploidea and in bootii. It varies a lot. In some populations, 100% of the individuals, the father was intermedia. So there's asymmetry in this group. Intermedia is almost always the sperm donor. Okay. So why? Why is that the case? So the first thing is this antheridigen system. Remember, I went back, talk about that just a second again. Not all species have this. It turns out not all Gyropters have this. And when we looked at this group, intermedia had a very strong antheridigen presence. but Carthusiana and cristata lack those, right? So intermedia is the diploid. These are the two tetraploids that are involved in that hybrid formation. Here we have Carthusiana, intermedia, triploidia, cristata, bootii, right? So intermedia had a very strong antheridigen system of itself and of these other two. Carthusiana does not respond to itself, nor does it respond to tritium. You cannot make male Carthusiana or cristata using this method. However, Carthusiana would convert intermedias to males, the cristata would convert intermedia to male, and the intermedia would convert itself to male. Does that make sense? It just likes being male for whatever reason, okay? So there's a variation in the presence of the antheridigen, and most of the spores on the landscape out there are going to be intermedia that are producing the sperm, because if you have two gametophytes together and one is intermediate and others one of these other two, is very likely going to convert intermedia to male. So there may be a preponderance of sperm coming from intermedia. So now let's look at there's a lot of data on this slide. So let me walk you through it. This is, we'll look first at longevity. This is proportion of sperm swimming. This is time. A little bit lower than us, but here we have intermedia, the diploid. Okay, here we have cristata, and here we have Carthusiana, the two tetraploids. This is per sperm longevity over time. You see that intermedia sperm live longer in general relative to either of the two tetraploids. So not only are they probably making more sperm out in the landscape, they are significantly longer lived than either of the two other parents that it's hybridizing with that are almost always the female donor. In addition to that, if you look at speed, intermedia is significantly faster. The sperm swims significantly faster than either of these two. Again, that's cristata in blue and Carthusiana, the two tetraploids in red, in green. So longer lived, faster swimming sperm. And if we did the calculations on the sperm, the time they could live and the swimming speed, we calculated the intermediate could swim about 12 centimeters. That's what is that, 12 centimeters? Whereas these other two were significantly less able to swim that distance, right? So there's something different about the sperm of intermedia, and which is sets it apart. So you can maybe start seeing, like, wait a minute, I can understand how this could be the male contributor in almost all cases. And then we did something that we did dig into the literature to try to find evidence of why this might be happening, other aspects of the archegonia sperm morphology. And there was a paper published in 2000 by Zhang et al. Where they hypothesized through no measurements that many polyploids or tetraploids are larger than diploids. Not always the case, but that can be the case. And they said, tetraploid sperm are 4 times as large as diploids, and so they just don't fit, right? And so here's the example of what I mean by that. These are the archegonia. I haven't talked much about archegonia, the neck sticking out. What happens in ferns is when these things get wet, the cells in the center dissolve and they open up, and the egg is down inside there. So the sperm have to swim around and go down in this neck and fertilize the egg that's embedded in the thallus of the gametophyte. So they hypothesize that tetraploid sperm are too big, they don't fit. We didn't find that. In fact, we found that, well, the first thing we found is that this is neck canal diameter of the archegonial neck. The neck canal diameter of intermedia was significantly smaller than either of the two tetraploids. So it is smaller, the opening is smaller, but the sperm of intermedia were smaller than either of the sperm from intermedia, from cristata or Carthusiana. So intermedia has small sperm, smaller archegonial necks, but look at the size of this sperm here. This is, what is that, 2.5, this is 4. Look at the opening size, 10. There's no way, this sperm can fit, and intermedia can fit in here, and these two sperm can fit in intermedia. So it's not a morphological reason why the sperm obviously can fit, based on the types of measurements we did. So that wasn't a really good hypothesis preventing the fertilization, or supporting the idea that intermedia is always the male because there's some mismatch between the gamete size. That didn't turn out to be the case in this study set. So why is Dropterus intermedia consistently the sperm donor? There are a lot of things that influence sexual reproduction in these plants, for sure, right? And I don't pretend to have all the answers, but we know the intermedia has a strong antheridigen system, so there'll be a lot of any late arriving spores of intermedia coming out there. If there are any other gametophytes, they're probably going to become male, saturated with males. I didn't talk about breeding systems. No, I won't talk about that. They have a less flexible breeding system, so just trust me on that. They, in both of the two cases of Carthusiana and cristata, they can self. I told you that didn't happen. They can actually self. I was talking with you about a few minutes, they can do that, but intermediate can't. So it's less flexible breeding system. And they have sperm that swim faster, live longer, and they're smaller, which the size may contribute to this in some way, but they definitely have some interesting aspects of their ecology, the physiology and morphology of the sperm that may play a role in understanding the mechanism behind all this asymmetry. I don't know. I mean, we'll see if that holds the test of time. So this really is just the beginning. That paper with Wes and Dave Barrington at Vermont was really an exciting paper we worked on a couple years ago. But we can continue to work on this project going back to the tropical ferns and now doing some stuff with Dryopteris, looking at all sorts of things. What are the impacts of pH? Huge impacts on pH, and whether or not the impacts of pH influence longevity and fertilization ability, temperature, water stress, and hopefully eventually have enough species to look at phylogeny, and then their ecology. What really started this project, I spent my whole career really studying the physiological, ecological ecology of epiphytic fern gametophytes versus terrestrial fern gametophytes. I built this whole array of data set, understanding why the morphologies vary, why they grow where they grow, and I was like, I really want to understand the physiology of the sperm. But it's just difficult to get this project to work. And so we don't have a big data set yet for that, but a lot more data are being derived from this slowly but surely. And with that, I'm happy to take any questions that you may have. Yep. Yep.